Hi everyone, my name is Miss Quinn and I'm a high school biology teacher in Canada. I'm so excited to be recording this video today about glycolysis because there has been a paradigm shift in our understanding of glucose metabolism, and I'm excited to explain it to you today. Sometimes this happens in science, a paradigm shift. It's when there's a fundamental change in our understanding of a concept. In the case of glycolysis, there's been a pivotal change to what we think is happening when glucose is broken down by our muscles for energy. George Brooks, that's this guy right here, he's a researcher in Berkeley, was the first to really question our conventional thinking about lactate in the 1980s when he first published his work on the cell-to-cell lactate shuttle. Today, Brooks's work is solidified in the scientific community. His research has been extensively peer-reviewed, replicated, substantiated, all the things. And while there's always more to learn, there's no question that our traditional teaching of cellular respiration is obsolete and outdated. I'm not really sure why it's taking so long for these new ideas to reach the classroom level, but I'm hoping to be part of the conversation in setting the record straight. The lactate shuttle theory changes some of the basic metabolic reactions we are taught in school. And these new ideas will need to be integrated not only into textbooks in high school, but also in research, medicine, and nutrition. So, let's go. Here's a diagram of glycolysis as it's traditionally taught in school. We see glucose entering the cell, and it's enzymatically broken down in a series of reactions we call glycolysis. Of course, the end product is pyruvate. In this reaction, we also generate 2 ATP, and 2 NADH. In this model, the fate of pyruvate is determined by oxygen levels in the cell. We have always taught that in aerobic conditions, pyruvate enters the mitochondria, forms acetyl-CoA via the link reaction, which can then enter the citric acid or Krebs cycle, until finally we have the electron transport chain to generate a huge quantity of ATP. That pathway is highlighted here in blue. We've told our students that in anaerobic conditions, like an all-out 400-meter sprint, our need for oxygen is greater than what can be provided by our breath. Our muscles become anoxic, and our cells resort to what we call lactic acid fermentation. In this pathway, pyruvate is converted to lactate. I'm coloring it red. We usually use lactate synonymously with lactic acid, and we've said that that lactic acid is a metabolic dead-end product that needs to be cleared. We've also explained that this lactic acid can cause acidosis in muscles, which explains that burning sensation we've all experienced in our muscles during intense exercise. Today, we have a much better understanding of lactate metabolism, and there's two main problems with our conventional thinking. The first is that lactate is always found in our blood. In a healthy resting human, we would measure 1 millimole of lactate, per liter in our blood, in our muscles, and our arterial blood. This is 10 times more than the levels of pyruvate that we find in our resting levels. If lactate is only produced in anaerobic conditions, then why is it found in our blood all the time in levels higher than pyruvate, which is supposed to be the main product of glycolysis? This tells us that lactate is produced in fully aerobic conditions, and we have the evidence to back that up. I'll provide a link to the study, but studies have been done on people exercising to high intensities, and while their lactate levels do increase, their oxygen levels are measured to be relatively stable. In other words, hard-working muscles do not run out of oxygen faster than ATP can be supplied. Our cells do not become anoxic or anaerobic. It's here where I tell my high school students that we are not microbes fermenting our muscles when we work out hard. I'm going to circle back to lactic acid at the end of this video and explain why it doesn't actually exist at all in our body. But for now, let me go ahead and explain our current understanding of glucose metabolism. In this model, we still begin with glucose entering the cell, And through the process of glycolysis, it's enzymatically broken down to again produce 2 ATP, and 2 NADH. However, you'll notice one key difference. In this view, pyruvate is immediately converted to lactate. This explains why it's measured in our blood in aerobic conditions. Lactate itself is the main product of glycolysis. If we were to zoom in more carefully to this reaction, we would see an enzyme here called cytoplasmic lactate dehydrogenase, which is converting pyruvate to lactate. In this process, it is oxidizing NADH back into NAD+. Lactate can now enter the mitochondria in the step here. There's something called an MCT transport protein here in the cell membrane. Lactate will be converted back into pyruvate. There's an enzyme called mitochondrial lactate dehydrogenase in the mitochondria that does this, to then become acetyl-CoA via the link reaction and enter the Krebs cycle as before. This is extraordinary. This tells us that lactate is the link between glycolysis and mitochondrial respiration. It's not a waste product we need to get rid of. It's important. But the more incredible finding perhaps is that lactate itself has been found to be shuttled to other working muscles, bypassing the need for those receiving muscles to do glycolysis at all. So let me break this down with a picture. Here we have two muscle types, a type 2 muscle fiber, which we also call fast twitch, glycolytic, or a white muscle fiber, and a type 1 muscle fiber, which we also term slow twitch, oxidative, or red muscle fibers. During times of intense exercise, like that 400-meter all-out sprint again, glycolysis starts in fast twitch muscles to generate a quick burst of ATP to power our movement. Lactate is produced as a product via glycolysis in these cells, and that lactate can then be shuttled to more oxidative muscles with more mitochondria, which will use that lactate directly as a fuel source for the Krebs cycle. For the receiving oxidative muscle, lactate has become the preferred fuel source, and we have uncoupled glycolysis from oxidative phosphorylation. In other words, this type 1 muscle fiber does not have to do glycolysis first. It can, but it doesn't have to. I should add, too, that glycolytic muscle fibers still have mitochondria and still do oxidative phosphorylation, but their main role is to export lactate to other cells and organs, including the heart and the liver. So, to recap, this represents a pretty big departure from our previous understanding. Lactate is produced in fully aerobic conditions. Lactate is not a metabolic waste product. It's the link between glycolysis and oxidative phosphorylation. And lactate can be shuttled to other cells and organs where it can directly enter the mitochondria for oxidative phosphorylation. To wrap up, I want to explain why lactic acid does not exist in our body. Again, people are not microbes. We do not ferment. Here's a diagram of the molecule lactic acid. It can be called also 2-hydroxy. Let me label my carbons here. 1, 2, 3. So it can be called 2-hydroxypropanoic acid for its hydroxy group on carbon 2. And of course, it has this carboxylic acid functional group that can donate a hydrogen ion, making this an acid. It has been wrongly assumed that the carboxylic acid intermediates that form during glycolysis and lead to pyruvate or lactate are donating hydrogen ions and acting as acids, both pyruvic and lactic acid. But this is not the case. I'll link a scientific paper that explains it in way more detail. But basically, the first time we produce a molecule in glycolysis with a carboxylic acid structure happens during step 6 of glycolysis. I've written it below here. In step 6, we have 1,3-biphosphoglycerate phosphorylate ADP. While the product 3-phosphoglycerate, that's this one here, I'm putting a blue bubble around it, has an ionized carboxylic acid functional group, there was never a hydrogen ion released here. It was phosphate. This 3-phosphoglycerate continues through glycolysis to become pyruvate and then lactate. But again, these molecules never existed as an acid. Neither pyruvic nor lactic acid were ever present. Interestingly, the conversion of pyruvate to lactate via LDH, that lactate dehydrogenase enzyme, consumes protons. So it's actually an alkalizing step. I'll just color that quickly here. You can see the 2 hydrogen atoms that make lactate from pyruvate. This is my lactate. This is my pyruvate. And there's the LDH enzyme. The step consumes protons. It doesn't produce them. So there's no acidosis here. All right, this is the end of my video. If you've made it this far, thank you for watching. I hope I've explained the ideas of the lactate shuttle clearly, and I'm glad to help shape what we know about glucose metabolism today.